Hi everyone, I'm Emily Richey. I am a student at the University of Arkansas-Fayetteville, and I am from Paris, Arkansas. My research project is entitled Effect of Proline and Hydroxyproline on Formation of Human FGF1 Amyloid-Like Fibrils. So my project studies neurodegenerative diseases such as dementia and how decreasing the amyloid-like fibril formation and aggregation can potentially help with preventative measures for diseases like this. So I study specifically human acidic fibroblast growth factor 1, which is a 16 kilodalton heparin binding protein that contributes to different cell processes throughout the body. In humans, when the amyloid-like fibrils of FGF1 begin to accumulate and aggregate, this is when you see neurodegenerative diseases like Alzheimer's disease begin to form. And so my project focuses on osmolytes such as proline and hydroxyproline and how they are believed to increase the solubility of these amyloid-like fibrils to help decrease their formation. And so if this is the case, then potentially proline and hydroxyproline could serve to inhibit the formation of the fibrils and thus help decrease the prevalence of neurodegenerative diseases like dementia. So I'm going to go ahead and talk a little bit about the primary molecules I used throughout my experiment. This is wild type FGF1, the protein I will talk about that I purified here in a second. And then as you can see, it has a beta trefoil structure. Over here, we have Thioflavin T. This is the dye I used throughout my experiments. It binds to the amyloid fibrils, so it really shows me when the fibrils are aggregating and forming. Down here, you have proline and hydroxyproline. These are the osmolytes I talked about that are the focus of my experiment. And then I have sodium dodecyl sulfate here, which mimics the structure of the phospholipid in the cell membrane. And SDS helps to induce aggregation in wild type FGF1. And so the purpose of using this in the experiment is to simulate what it would be like to have my experiment actually in the cell. And so having this mimic what a cell membrane would do in the experiment shows me a little bit more how my project can be applied to the human body. Okay, I'm going to go ahead and talk about a few of my different experiments I've done. Beginning with SDS page purification of FGF1, I used a heparin sepharose column and eluted my protein using different concentrations of NaCl. As you can see here in lane 1, this is my purified protein that was at 1500 millimolar NaCl, and then here in lane 2 is the marker. So this was already a purified FGF1, and you can see that the bands align, which tells me that I have a purified protein. This is my Thioflavin T fluorescence of FGF1 with different concentrations of both proline and hydroxyproline. Overall, the trend I see in both of these graphs is that as the concentration is increasing of both proline and hydroxyproline, we see a decrease in the fluorescence of wild type FGF1 amyloid fibril aggregation. And so this is really promising because if both proline and hydroxyproline are showing to potentially decrease this aggregation, then they could potentially help to reduce the aggregation in the future of amyloid fibrils, which contribute to neurodegenerative diseases. This shows the effect that SDS actually has on wild type FGF1. As you can see, the blue line is 0.2 millimolar SDS on its own. The black line is just wild type FGF1, and the red line is wild type FGF1 in the presence of SDS. So you can see how drastically the fluorescence increases, which shows how SDS actually induces aggregation. Over here is the UV-Vis absorbance, which shows me the turbidity, which tells me a lot about the aggregation as well. And so I combined wild type FGF1 and SDS, which shows me, the induced aggregation in wild type FGF1. And then I bring in proline to show how proline reacts to this aggregation. And so this experiment overall, you see it's starting to decrease. The aggregation is. And so down here around 3.5 to 4 molar proline is where we see the absorbance at the lowest. And so this suggests that these concentrations of proline may serve to reduce aggregation the best. Okay, I'm going to go into my third column here. This experiment of fluorescence of FGF1 with different concentrations of proline really echoes my last experiment. As the concentration of proline is increasing, we see an overall downward trend of the fluorescence, but specifically at 4 molar, we see the lowest concentration, or we see the lowest fluorescence. And this shows that 4 molar may serve to be the best at decreasing the aggregation. So in conclusion, both proline and hydroxyproline might work to decrease the formation of the fibrils in the body, and potentially hydroxyproline may actually do it more effectively since it is able to decrease it at a lower concentration. This is something I'll continue to study. And you've seen how I incorporated SDS to see how this will work in the body. So I'm really excited about the results I've had so far. And I hope to do a circular dichroism spectrophotometry experiment soon to study the secondary structures of wild type FGF1. Thank you for watching.